Why should anyone care about biurate protein experiments? Well, the answer lies in the very fabric of life itself. Proteins, you see, are the building blocks of life. They play a crucial role in almost every biological process, making their detection and quantification vital in many scientific experiments. One of the most commonly used methods for this is the biurate protein assay. Imagine a world where you can identify the presence of proteins and even measure their concentration. That's the world of biurate protein experiments. This method is named after the biurate reagent, which turns a distinctive shade of purple in the presence of proteins, making it easy to detect them. In the first step of a biurate protein experiment, you start by preparing the biurate reagent. It is a concoction of copper sulfate, sodium hydroxide, and potassium sodium tartrate. This reagent is the magic potion that will react with the proteins to give a color change. Next, you add a known volume of the protein solution to a test tube. This solution could be from any source, a cell, a tissue, or even a food item. Once you add the bioreagent to this, you mix it thoroughly. Then it's time to wait. The reagent takes a little time to react with the proteins, and after about 20 minutes, you should see a change in color. If proteins are present, the solution will turn purple. The darker the purple, the higher the concentration of proteins in the solution. Not only does the color change indicate the presence of proteins, but it also gives you a way to measure their concentration. By using a colorimeter or spectrophotometer, you can measure the intensity of the color and hence the concentration of proteins. This method, simple as it may sound, is a powerful tool in the hands of scientists. It allows them to study proteins in great detail, understand their structure and function, and even design new proteins for specific purposes. So to summarize, the biurate protein experiment is a method used to detect and quantify proteins. It involves preparing the biurate reagent, adding it to the protein solution, waiting for a color change, and then measuring the intensity of the color to determine the protein concentration. It's an essential tool in biology, biochemistry, and many other scientific disciplines. And understanding it can open up a whole new world of scientific discovery. So the next time someone asks, why should they care about biurate protein experiments, you'll have the perfect answer. It's all about understanding the very fabric of life itself.